Okay, so today we're going to be looking at the DNA portion of the May-June Senior Certificate National Senior Certificate Examinations of 2025. We're going to be looking at it, we're going to be dissecting it. We're going to see what was a little different with this year's paper compared to previous year's papers. So without any further ado, let's get straight into it. So we're going to start at 1.4. Obviously, before this was multiple choice, biological terms, A only, B only, none, both A and B. We're not going to look at any of that. You're going to jump straight into the DNA portion of things. So it says here the diagram below represents two biological processes occurring in the nucleus. Now, firstly, here, when we see the word nucleus or when we hear that it's occurring in the nucleus, automatically we should associate two important biological processes with the nucleus, and that would be DNA replication based on our extensive knowledge at this point, and transcription. We should know at least that, right? That those two processes are taking place in the nucleus, right? So automatically we know it's going to be one of these. Now, when we're looking at this one here, if we look at this specific portion here, the X molecule, right? The giveaway here, or for me personally, there might be other giveaways for you, but the giveaway for me personally, let's use indigo. The giveaway for me personally would be the presence of use, right? So automatically that would tell me that this is transcription. Oop. Let's just rewrite that. Transcription. There we go. That will automatically tell me, okay, this is transcription. Now, if we go a little lower, what would also give it away here is that if we look at, okay, let's first do diagram 2. Diagram 2 obviously now would be DNA replication. The reason being is we have T's present, but also we have two strands taking place, right? We see the formation of two strands here. So that would be that portion that would tell us, okay, no, but this is DNA replication, matriculants. Also, what I want to urge you to do is to not leave this DNA out here and just write replication. We don't know what happens at finals. We don't know if they will just accept replication. So please just do write DNA replication, right? Because the argument can come across, but someone will write RNA replication. So just be specific and write DNA replication, please, dear matriculants. I implore you. So we've now discussed that, so we know that diagram 1 here is transcription, and we can know that diagram 2 now is DNA replication. They then go and ask us to identify molecule X. Now molecule X, because it's transcription, will be the mRNA, right? We've now discussed that, so it must be the mRNA. Also, we see the presence of U's there, which also tells us it is the mRNA. What further tells us it's the mRNA is it looks like a comb, right? It always looks like a comb. It's single-stranded, which also tells us it is a form of RNA, right? It's not double-stranded like DNA has the tendency, or, you know, RNA looks like that, DNA looks like that, right? So that would be, see, there's several indications as to why that would be considered. Let's just erase as to why that would be considered the mRNA. So let me just erase so that for the next couple of questions, I think I need it to be nice and clean. Alright, let's move down. They say, give the natural shape of molecule Y. The molecule Y, we obviously also know now this is DNA, right? So molecule Y's natural shape here is double helix, right? It's not double stranded. or single-stranded. When they're referring to natural shape, we're referring to helix structure, the helix structure, right? So double helix would be the answer there. Then they go on to say the phase of the cell cycle during which diagram 2 occurs. So they want to know, when does DNA replication occur? Now DNA replication occurs in the interphase, right? DNA replication occurs in the interphase. So I'm not going to be explaining too much on this one. I'm just going to be taking you through the answers. But we have a cell division taking place here, and we have a cell division taking place there. Cell division A, cell division B. The period between these two cell divisions here is what is known as the interphase, and it is in this interphase that we see that DNA replication takes place. It is in this interphase period, right? DNA replication takes place in the interphase, right? They say the full name of the nitrogenous base that is found in molecule X but not in molecule Y. So we now know that molecule X is the mRNA, and we know that molecule Y is DNA. Now in DNA we have thymine, in RNA we have uracil. So the answer for this one here would be uracil, because uracil is found in the mRNA but not in DNA. Right? These are easy questions. The sugar found in molecule X. Now the sugar found in molecule X, people, you can't just write sugar because they already tell you it's a sugar. So you need to be specific as to what sugar. Look, when we're looking at DNA, we have a certain sugar. Now with DNA, the sugar is called deoxyribose. Right? Sugar, it's deoxyribose sugar. Deoxy meaning there's a lack of oxygen, right? And then RNA here means that, RNA is a ribose sugar, so it does not have a lack of oxygen. That's just for interest sake. But the answer here is that this is a ribose sugar. Right? That is a ribose sugar. Then we look at bond Z. Bond Z, now the bond here between two nitrogenous bases is always a weak hydrogen bond. It is a weak hydrogen bond, right? A weak hydrogen bond. If you, for example, and I will always reiterate this point, if I will always suggest writing out the full word. If you want to write weak, now some people are going to think H2, that's fine. But some people forget that it's not like that. That's wrong, right? That, I don't even know what that means, honestly. It's like that, right? So let's just be specific. So let's rather use words instead of chemical symbols, just so that we know we get the full amount of marks. We don't leave anything to chance, dear matriculants. They say state the number of codons on molecule X. So that would have further given us, that is why reading time is so important, dear matriculants, because this word codons over here. Oh, wrong one. This word codons over here. That codons is for mRNA. So maybe you didn't know it was mRNA, but you know that mRNA has codons. So if you read, you would understand that if they're saying codons for molecule X, you would know that at 4.2.1, yeah, it is the mRNA. So that was nice for them to put that in there. But anyway, they say state the number of codons on molecule X. So when we're looking at codons of molecule X, now before I even do that, what I need you to understand here, and please never forget this information that I'm sharing with you now. When they're referring to the DNA-based triplet, when they're referring to the codon, when they're referring to the anticodon, right? When they're referring to the anticodon, I need you need to be very, very careful as to how you count. When you are counting these things, you're going to always count in sets of 3. Sets of 3. You count like that. Sets of 3. Because, and I think it's going to be picked up now when they mark. If you don't know, certain learners are going to say that there is 3, 6, 9, 12 codons. But that is completely and utterly wrong. Right? There is 4. So we count 3, 3, 3, 3. So there is 4 codons, right? 4 codons, not 12. There is 4, right? There is 4. There is 4 codons. So that was this question. Very easy, very simple. Okay, this is a genetics question. This is a nice question. This is a dihybrid, I think. Yes, we have, yeah, this is dihybrid. This is nice. Look at that a little bit, not in this video. And we're not also going to be looking at this. I want us to get to this question over here. This is a nice question over here. Let's just look. They say the diagram below represents a stage of protein synthesis. So a stage of protein synthesis, we know that there are 2 stages of protein synthesis. You have your transcription and you have your translation, right? 2 processes. They say name the stage of protein synthesis represented in the diagram. We will know that this is translation, and I'm going to tell you now how you identify that this is translation. It's simple. You identify it because of the presence of the tRNA molecules. That is how you will identify that this is transcription, because the tRNA molecules are present. It would be, translation, because the tRNA molecules are present. So we know that it is because we have tRNA molecules that are present, right? tRNA molecules are present. So this is translation. Then we also know the site where this process named in question 2.3.1a occurs. So translation doesn't occur in the nucleus. It's only DNA replication and transcription that occurs in the nucleus. Translation occurs in the cytoplasm. It occurs in the cytoplasm, right? They say describe the role of molecule X in protein synthesis. So they're asking us, what is the role of tRNA in protein synthesis? Now, this comes from what is translation, right? If you know what translation is, you will know what, then you will automatically know what the role of the molecule X is. Now, what they like doing this because, I just want to quickly state something, they like doing this because of the fact that they don't always want to give you that 6 marks. You know, sometimes it is for free in most cases. So what do you know about tRNA? We know that tRNA carries a specific amino acid so that the anticodon can match the codon, right? So that it can bring the required amino acid to the ribosome, right? So basically, tRNA's function here is you just say it carries a specific amino acid, so it carries a specific amino acid, right? It carries a specific amino acid, and it also brings the required protein, right? It also brings required required amino acid to ribosome, right? So that would be the functions of the tRNA, or the role of the tRNA. Then they say identify the complementary bases from left to right for anticodon 2. So anticodon 2, okay, we're going to stop here because I am running out of time. So we're going to stop just here, and then I'll come back shortly. Alright, so let's just quickly get back to the last thing we stopped at here was we discussed here the role of the ribosome, the role of the tRNA, sorry, I'm looking at the word ribosome, the role of the tRNA, and we said here the tRNA carries a specific amino acid. It brings the required amino acid to the ribosome. So we discussed those 2 points in depth. So now what I'd like us to do is we're going to do 2.3.2, and then we're going to do 2.3.3 and 2.3.4. So with 2.3.4, especially please do pay attention. It is not the easiest question. So that is why I'm making a separate video or a part 2 of this question. So we can see with 2.3.3, they say identify the complementary bases from left to right. So left being this side, right being that side, representing the following. Anticodon 2. So here, this here is the codon, right? Because the structure here is the mRNA. So they want the anticodon, so they want these 3 over there. So the anticodon C is going to bond with G, A is going to bond with U, and U is going to bond with A. So you'll have GUA. Then they want the DNA triplet for codon 1. So if we look here, the DNA triplet for codon 1. So we have the anticodon, which is CAU. The codon of that would be GUA. And the DNA triplet, G will always bond with C, U bonds with A bonds with T, because we're in DNA, so we cannot have a U, so that is going to be GAT. Then they go further to say that, so this is 2.3.4. They say the table below represents the anticodons coding for different amino acids. Anticodons, tRNA. Right? Now they say a mutation occurs during replication. So I already did this question. That's why you'll see I wrote on here. That's why you'll see this circles. They say the mutation occurs during replication. Right? The mutation occurs during replication. That's our first key important. It's where it took place. It took place in a replication. Then it says the mutation results in the amino acid proline being replaced by arginine. So they're kind enough to tell us what is being replaced. So they're telling you here, proline is replaced with arginine. And then they say use the table and explain how the mutation will affect the protein that is synthesized. And it's a 7-mark question. Now what is fascinating about this is that we're not normally used to having this type of 7-mark question. But it's not a difficult question. The first point here, okay, let me just analyze a little bit further. Also we know that proline is replaced with arginine, right? And they want us to end up by the protein part. Right? So obviously there's no memo for this question yet, so I'm doing it based on my understanding, and I assume my understanding is correct. I don't know if there's here we go. There's an empty space for us. So we know proline is CCU, and so let me just write this down. So we know that proline is CCU and arginine is CGU. So obviously we can see we can already see that the C and the G are obviously that's where the mutation occurred, right? But remember, this is the anticodon, right? And they said that this occurred in replication, which means that you need to go back to the DNA base triplet and explain from there. So you need to first now translate to a codon and then transcribe to the DNA, right? So it's going to be GGA, right? This is now the codon part, and this is going to be GCA, and then this is going to be CCT, and this is going to be CGT. So we can automatically see here we have a C and we have a G. So now you need to explain from here what is happening, right? So if I'm just gonna erase all of that quickly, because now we know what is the DNA base triplet for both, right? So with that information we can do a lot now. We can actually answer the question, because remember they said it happened in replication. So we're gonna first say where did it happen. So it said it happened in replication. So in DNA replication, you need to say in DNA replication what happened. So in DNA replication, the DNA triplet, right, for proline changed from CCT to CGT as a result of a mutation. I mean, we got it from the question, right? Obviously we just put it during DNA replication. So we need to say what happened. Then from the DNA unit I explain the codon now. So you need to say the codon, or we can, let's be specific and say the mRNA codon. The mRNA codon, because why we have to do it like this is because they telling us, let me just quickly just touch on it again. They telling us it started in replication and we need to explain how it's going to affect the protein. So we must go from the replication point all the way to the protein formation. That is why we need to do it from the DNA, because you can't use tRNA or RNA or anticodon and codons in DNA in the replication, right? So the mRNA codon changes from GGA to GCA during what process? During transcription. Then you need to now, so now you've done the DNA, you've done the mRNA, now to do the tRNA. So now you need to say the tRNA anticodon so the tRNA anticodon changes, you can say changes, or let's not say changes. Let's maybe first say what happens first. So let's say the tRNA codon, yeah, let's make it changes. Changes from CCU to CGU, right? Where CCU would have brought proline, but now CGU brings arginine, right? It now brings arginine. So I just want to explain why I'm doing it this way. Firstly, I'm saying what change took place, right? I'm firstly saying what change took place. Now I'm saying if it was still CCU, if it was still CCU, it would have given us proline. But it changed from CCU to CGU, and it now brings arginine. Alright? And the reason why I'm only speaking about the amino acids here is because in the table it was anticodons with amino acids. If it was codons with amino acids, then I would have spoken about the proteins at the codon. If it was the DNA, I would have spoken about it there. Right? So that is what I'm trying to bring across here, right? That's why I'm only speaking about amino acids by the tRNA, because that is the codons. Because in the table it was the codons, right? So that would be like maybe the first 3 marks, in my opinion. I don't know how they're marking it yet. Alright. Now you need to say what is going to happen because of all of this. Right? Now because do I have, oh, there's no more free spaces. Okay. So I'll write here. So now, because of that change in from DNA to the tRNA, what happens is arginine is now obviously going to replaces proline, right? So arginine replaces proline where in the polypeptide chain. You always want to say what happens and where it happens. What is this going to cause? Because of that change, it's going to alter the amino acid sequence. Remember, there's a sequence. And this may, emphasis on may, change the protein this may change the protein that must be formed. So anyway, must be formed. Last point I'm gonna put here. Right? This may cause protein to become non-functional or less efficient, or let's say harmful. Put it like that. So I'm going to explain again from the beginning. That's why I knew this was going to take some time. So they say the table below represents the anticodons coding for different amino acids, right? But remember, the important part here is that the mutation occurs during replication, which means we need to transcribe the anticodon, translate it back to the DNA base triplet, which I can see I've done there. Right, and then we need to explain from here right down, right, for each part. So we can explain at the DNA what happened. Right, we can see the DNA triplet change from double CT to CGT. Then the codon changed from GGA to GCA. Then the tRNA changed from CCU to CGU. Now the important part comes in here. You don't just leave it at changes, you need to say what amino acids, because in the table it was specific that the anticodons match the amino acids. You can't be speaking about amino acids here because it's not here. It's not going to be the same. Right? So now you can say CCU would have brought proline, but now it is going to bring arginine. And then we say, obviously now arginine is going to replace the proline in the polypeptide chain. And this will alter the amino acid sequence. And this may change the protein that must be formed. Right? So, because arginine, remember, if you have amino acids like that, right? Each one is different. And therefore, that protein can become non-functional if one is wrong, and that is